So I'd like to introduce our first speaker for this session. It's Professor Peter Donnelly. Professor Donnelly is also the director of the Wellcome Trust Center for Human Genetics, as well as Statistical Science Fellow of St. Anne's College. He is well known for his work in molecular evolution, tracing the roots of human existence to their earliest origins using the mutation rates of mitochondrial DNA. And he studied genetic distributions in living populations to trace human evolutionary history, which informs research in evolutionary biology as well as medical treatment for genetic disorders. He's a key player in the international HAP-MAP project, an ongoing international effort to model human genetic variation and pinpoint the gene responsible for specific aspects of health and disease. Also, he's a leading expert on DNA analysis and the use of the forensic science in criminal trials, and is an advocate for bringing sensible statistical analysis into the courtroom. And his topic today is the genetics of personalized medicine, challenge and opportunity. Professor Peter Donnelly, please.
Thank you very much. It's a great pleasure to be here in Macau, and it's an honor to have been asked to speak at such an exciting and interesting symposium. I'd like to take the opportunity, first of all, as Simon did, to acknowledge and thank the support of the Dr. Stanley Ho Medical Development Foundation, and in particular Mr. Patrick Hyun, for all of the work we've done in Oxford for the Centre for Personalised Medicine. Simon talked a little bit about that work within Oxford. It's a fantastic opportunity for us to help various groups, various stakeholder groups, students and clinicians, patients and members of the public understand genomics and personalised medicine, and we're extremely grateful for the very strong support, and I think it's fair to say the visionary support of the Foundation in order to enable us to do that. But much more than that, as you've heard already, that support has also facilitated our links already with the Chinese University of Hong Kong, with Professor Joseph Sung and Dean Francis Chan and their colleagues. We've had 2 symposia in Hong Kong, the second one of which was yesterday. We're very fortunate in benefiting from the advice of Professor Dennis Lowe, who's a member of our External Advisory Board, and his insights have been extremely helpful over the 4 years of our Centre for Personalised Medicine in Oxford. That's something we're excited about that's already happened. We're absolutely thrilled, as you've heard from Patrick. We'll have discussions today about the possibility of extending that to collaborations with the University of Macau, and that's something we're really, really excited about. And again, very grateful for the opportunity. So the Foundation support has enriched our professional activities, and we're very grateful for that. I also want to say, personally, they've enriched my life. I'm very grateful for Patrick's friendship and for Isabel's friendship and their family, which has been a wonderful thing over the last few years. I want to talk again about genetics and genomics and the ways in which it can impact on healthcare. And in a sense, all the things I'm going to say are rather complementary to what Simon talked about in the previous lecture. He talked about cancer. As he said, cancer is fundamentally a disease of the genome. Cancer cells have messed up genomes, and that's what causes the problem. So it's a very natural setting in which to use genetic and genomic techniques to try and develop particular treatments. And you've heard a lot about the journey that the field has been on and the progress that's been made. I want to talk largely about how genetics and genomics can help in non-cancer diseases in a number of different ways. And I want to give you, I'll give you a number of examples that'll give you a sense of the potential, I think, both for genetics and genomics to make us much better at developing drugs, at developing drug treatments, and also make us much better at understanding individuals and using mathematical and statistical techniques to do a better job of predicting risk for individuals and working out what the right interventions are for particular individuals. Now, Simon mentioned that the sequencing of the first human genome cost about $3 billion. So there are 3 billion letters in our genome, our DNA code that's contained inside every cell. That Human Genome Project, that the completion of which was announced in 2000, that took $3 billion. It took thousands of scientists in many countries, the UK and the US, but there was support from China and from Japan and from a number of other places as a key part of that international symposium. Well, happily, we've got better at sequencing human genomes. There are much cheaper and more efficient methods, and the costs are now, as Simon said, under $1,000 per individual. So this is a picture that shows not the decreasing cost of human genome sequencing, but the increasing numbers of individuals who have been sequenced. It's already a number of years out of date, and I don't know what the exact numbers are. I suspect on the planet as a whole, there are maybe 100,000 or 200,000 individuals who have had their entire genome sequenced. It's easy to see a path for that number to become millions in the next few years. This is a figure from a paper that made projections based on various assumptions, and those projections differ a little bit. But my own view, and it's consistent with some of the projections and that of others, is that within about 10 years, and certainly within 15 years, there will be 1 billion people on Earth who have had their genome sequenced. 1 billion people. And for many of those, that information will be linked to information about them, either through linkage to electronic medical records, or increasingly through the information that's collected about us through wearables, through our watches and our phones and similar devices. And that, I want you to get a sense, that presents an enormous opportunity. There are challenges. It's a ridiculous amount of data. So big data is all the rage, and different fields compete for how their field produces more data than someone else's field. However you measure data, whether it's the amount that's generated, the amount you need to store it, the amount of computer power you need to process it, however you measure it, genetics and genomics and its connection with medical records will be, in 10 years or so, one of the biggest data areas that we're faced with. So there are lots of opportunities. What are the challenges? Well, it's been mentioned already. The challenge is that Simon talks specifically in terms of cancer, but it's true across the board. It's probably even more true across the board. We are not very good at developing successful drug treatments. So the left-hand graph, as you look, shows the declining number of successfully approved drugs over the years, and the right-hand side has a number of figures there. The first one, that figure of 11.8%, about 10%, that's the probability. So when a pharmaceutical company is developing a drug, they do a huge amount of research initially. They use that research to choose the target of the drug, which bit of our biology is the drug trying to change. And then, totally appropriately, there are a series of trials, regulated trials, that need to be undertaken to check whether the drug, first of all, to work out what the right dosages are, then to check whether the drug is safe, and also whether it's effective, whether it's any better than what the existing standard of care is. So, in spite of all the research that goes into the preclinical work, the success rate of drugs, so once a drug goes into a clinical trial, what proportion of them succeed? The answer, very depressingly, is about 10%. So 90% of drugs don't get through the clinical trial process, either because of concerns with safety, or quite often because they just don't work. They're not any better than what's out there already. That process is extremely expensive. It's estimated to cost somewhere between $1 and $2 billion to go all the way through the clinical trial process. And if you're doing something that is extremely expensive and you're failing 90% of the time, then the times you do succeed, you need large revenues to recoup all of those costs. And that's one of the key things at the heart of the cost of current expensive costs of drug therapies. It's because, as a field, we're really bad at choosing drug targets. Why might that be? I think there are lots of different factors, but I think one of the major ones, and it's been compellingly argued here by a number of colleagues, is that all of the preclinical, well, virtually all of the preclinical work that's done, the stuff that leads to the choice of a particular target as an intervention, that's necessarily not done in humans. It's done either in what we would call model systems, either cellular models or animal models of various kinds. And while they are extraordinarily important for our understanding of biology, it's sort of obvious in one sense, but also very clear in another, that animal models simply don't replicate everything about humans. So the problem we have, and it's been argued, I think, convincingly, the reason we're so unsuccessful at developing new drug targets is that we don't understand human biology and we can't study it in humans. So one way of thinking about this is that when we have a new drug therapy, the challenge is that when we intervene, we don't know what will happen. And there are two different sorts of questions. And as a statistician, I think of one of these as sort of relating to the mean and the other one relating to variation. So at a basic level, the question is, what's the typical response to this drug? Will it tend to improve people who have a particular disease? That's like the mean effect. The other question is, how do I understand the individual variation? Because if you've heard from both speakers already, drugs work in some people but not others. So what are the factors that cause that variability? That's another key question. We don't know the answers to either of those. Why don't we know the answers? Because depressingly, we don't understand enough about human biology. Why don't we understand human biology? Because it's not easy to study. How do we normally do science? We normally do science by designing experiments. Those experiments involve interventions, so you change something and you measure what happens, and you falsify a hypothesis, or you learn more about the underlying situation. For very obvious reasons, we can't do those sorts of experiments in humans. So we have to study humans very indirectly in other systems. A key idea I want to get across is that genetics offers us an opportunity to get around that problem. So human genetic data linked to rich molecular and clinical outcome data allows us to understand something about human biologies in humans, and that's one of the things I want to explain during the presentation. But here's the key idea, and it's a very simple one. We've got 3 billion letters in our genome, and if you compare any two human chromosomes, yours with the person next to you, then those will tend to agree at about 999 positions out of 1,000 in the DNA. Actually, as an aside, if the person sitting next to you weren't a person, but they were a chimpanzee, your DNA and their DNA would agree at 99 places out of 100. So we're quite similar to our nearest animal relatives. So although our DNA is similar between people, there are lots of places at which they differ. So we all inherit a unique combination of genetic variation. Some of those positions, so those changes in our DNA, they cause things to change inside us. They change some aspects of our biology. So here's the basic and very simple idea. When we give someone a drug, we are trying to change an aspect of their biology. And we want to know what the consequences are. So the idea is, in order to understand what the consequences of the drug changing that bit of our biology is, find individuals who happen to carry genetic variants that affect the same aspect of our biology, and just look at what happens to those. So it's a little bit like nature happens, by giving them an A rather than a T at their DNA, nature happens to have given them a drug early in their life, or a weak version of the drug you're proposing, and we can watch what the consequences are, provided we have the right kind of data and the right kind of analytical methods. So it's a very simple idea. It's powered by data, so the more data we have on people, data about their genetic variation and then what happens, stuff we can measure in them, the more powerful. Almost uniquely, I won't have time to go into this in too much detail, but almost uniquely, genetic data about DNA sequences allows us to understand causality in human biology. So as a statistician, we often teach people that association is not causation. Just because two things are correlated, it doesn't mean that one's causing the other. And in human biology, most of what we can measure is about correlations. They're very helpful for prediction. They're not always helpful for understanding causality. And they're not always helpful, and typically they're unhelpful, for knowing how you should intervene, what should you change, because you need to know about causality for that. So genetics is really helpful in untangling that. But it requires serious and relatively sophisticated analysis. So I said there are two broad ways in which genetics can be helpful. One is to understand the typical consequence of intervening in biology in a particular way, of a particular drug therapy, and I want to give you some examples of that. And then the other one is to understand, help us understand bits about individual variation, and I'll give you some examples there as well. So these days I combine my academic work with work in a spin-out company that we created to take forward some of those ideas at the scale we think is needed to deal with the data and the amount of data that's currently available and will become available. The company is called Genomics PLC. It was founded by myself and a number of colleagues in Oxford, as I said, to take forward the sort of things we developed in our academic careers. So we brought together data that's already been collected, typically in research studies, and we were involved in some of these, and I'm sure many of the scientists in this room and our colleagues have been involved in them as well. We brought together data which is available from research studies, which measures genetic information in individuals and then outcomes about them. So those outcomes might be at the molecular level, which genes are turned on, and can we see differences in the amount of protein produced by a particular gene, so RNA sequencing or gene expression studies. What about the levels of the proteins that are present? How do those translate into changes into things we can measure in people, cholesterol levels, levels of various biomarkers, and so forth. And then to other things we can measure up about people, including health outcomes, whether they suffer from particular diseases, what the progression of those diseases is, and so forth. So within the spin-out company, we brought together data from more than 1,500 different studies. In total, they involve over 5 million different people, and we have measurements on more than 5,000 different things, all the way, as I said, from molecular phenotypes to health outcomes. And then, so one way of thinking about this resource we've created is, so there are 3 billion letters in the genome. Many of those don't vary or vary very infrequently between people. So if you look at the ones which tend to vary a little bit more, say 14 million of them, for any one of those 14 million, you can think of us as having a big matrix, so 14 million columns, one for each of these positions, SNPs they're called, that vary, and then 5,000 rows for each of the things that we can measure. And at any one of those positions, we can look and see that if you change an A to a C at your DNA in this position, what's the consequence of that on the expression of this gene, or the abundance of this protein, or someone's cholesterol level, or whether or not they get inflammatory bowel disease. And we can look at all of those effects exactly so that we can do the kinds of things I was describing, to use these genetic changes to mimic what might happen if we give someone a drug. So I want to swap now between this presentation and, rather bravely, a live demonstration. So this is, we have all this data, we've developed various analytical tools for looking at it, and I just want to give you a couple of examples to flesh out this idea. I'll give you a couple that we would think of in science as positive controls, so we kind of know the right answer, and it's just checking that if we apply the method that I've described, we get something sensible. So this is one place in the human genome. It happens to be on human chromosome 5 at position 74 million, etc., along the chromosome. And it's a variable position. Variable position, SNPs have names. This one's called, not very exciting names, this one's called RS12916. And it's near a gene, and that gene is called HMG-CoA reductase. So many of the clinicians amongst you will know that that's the gene which is the target of statin drugs. So those drugs have been around for 20 or 25 years now. They've had a substantial impact in lowering LDL cholesterol levels. And as a consequence of that, they've had a massive impact globally in lowering the burden of cardiovascular disease and lowering mortality from cardiovascular disease. So this is a case where actually we know about the drug and we know what it does. Now, what I'm going to show you is that if we use the idea I've described, if we find a genetic variant that does something that's a bit like the drug, and look in people who are carrying that's like they've been on a version or a weak version of the drug all their life, what can we see? So this particular genetic variant, humble name, RS12916, amongst other things, has the property that it changes expression of this gene, kind of dials it up or dials it down a bit in the liver. So statin drugs inhibit this gene, HMG-CoA reductase. That's like dialing it down a lot. The variant that's plotted here, the alternative allele, if you change from a T to a C, then that actually dials it up. So the way it's plotted here is in the opposite direction from the drug. And what does this picture mean? So each row here is one of those things we've got measurements for in people. In fact, we've got many different studies of cholesterol, and most of the rows you can see are about cholesterol. These circles are telling you what the effect of the change from the T to the C, so what's the effect of dialing the gene up a bit. And the bars show our 95% confidence interval. And so the fact that all of these circles and bars are away from the null hypothesis, the line that says there's no effect, are saying that if you look at individuals who happen to carry a genetic variant which dials the gene up a bit, does the opposite from the drug, then that increases their cholesterol levels. Or the converse of that is, if you look at the other individuals, the individuals who carry the variant which dials the gene down a bit, that's like individuals who've been on a weak version of the drug all their life, they have less LDL cholesterol. So it's one of those instances, as I said, we know now that the drug works, but had we had this data 30 or so years ago when the drug was being considered, we'd have been able to say, actually, this idea suggests the same thing. Most of these measurements are cholesterol levels. One of them is heart disease, coronary disease. We now know that LDL cholesterol is causal for heart disease. Again, 30 years ago, all that was known at that point was that individuals who had high levels of cholesterol tended to have more heart disease. That's this association and causation question. You can't tell from that whether cholesterol is causal for heart disease. That's one possibility. Another possibility is that as people get sick with coronary disease, something happens which increases their cholesterol level, so that way, in that instance, the causality goes in the other direction. Or maybe there's a third factor that's affecting both of them. So there are three possibilities if you see the correlation. Only in the first case, only if LDL cholesterol is causal for heart disease, is there any point in intervening to try and lower cholesterol if what you care about, as you do, is heart disease. Now, what this is showing, so we now know that LDL cholesterol is causal for heart disease. How do we know that? Well, we did the following experiment. We took two large groups of people who are otherwise evenly matched. We intervened in one group to lower their cholesterol level. We did that by giving them statin drugs in a clinical trial. We followed them over time, and where we had intervened to lower the cholesterol level, we saw a decrease in cardiovascular disease. We could do the same experiment and take two groups of people who are otherwise evenly matched, and some of them happen to have inherited this genetic variant, which lowers their LDL cholesterol level. They don't know that, but you watch them over time, they've got a bit less LDL cholesterol. What this data point says is they get less heart disease. So that actually establishes, or it's a key part of the argument, that establishes causality, that shows that LDL cholesterol is not just associated with cardiovascular disease, it's causal for it. One more example, again, partly a positive control, so this is a different genetic variant. It's called RS2228145. It's on chromosome 1 at position 154 million and something or other on the chromosome. And the two letters that people have on their chromosomes at this position are either an A or a C. So again, what does this variant do? It's near a gene called IL-6R, so that's the receptor for something called interleukin-6. It's a key part of the immune system. The receptor sits on the membrane, on the outside of cells, and it's used to signal information from the inside of the cell to the outside that the immune system uses. And there is a drug available. A number of companies have the drug. So Roche have a drug called tocilizumab, which blocks that receptor. It stops the receptor from working. And that drug improves outcomes for people with rheumatoid arthritis. So this genetic variant, again, is another example where it does something that's pretty similar to the drug. It happens to affect the way proteins are made and cleaved. And if you have the C variant here, then that decreases the amount of receptor you have on the cell membrane. So the drug blocks the receptor, so it means there's less that works. This drug means that this genetic variant means that there's less to start with, and so it's a bit like the drug. What do you see? Well, one of the things you see here is that those individuals who have the genetic variant that's a bit like the drug get less arthritis. So we know the drug works for arthritis. You also see they get less inflammatory bowel disease. They get less Crohn's disease, less ulcerative colitis. So that suggests this drug might also be useful for that condition. You also see they actually get less coronary disease. And there are trials underway at the moment to see whether intervening in this pathway by blocking the receptor will be helpful for cardiovascular disease. So it's another positive control. I've talked about two examples we can actually look across the genome as a whole. There are 20,000 genes in the genome, and you can use this idea. If I want to know what happens if I perturb the gene with a drug, I can look at genetic variants which affect the gene and see what happens to the people who carry those variants. So I've looked at one gene already, HMG-CoA reductase. So these pictures, these kind of circular pictures, they're a bit like car speedometers, but they go the other way around. So the fact that this red line goes all the way around the clock, backwards as it were, is saying that the genetic evidence says that if you perturb this gene, then you have an impact on conditions to do with lipids and heart function. And we already know that's true from statins. The next example is a gene called PCSK9, so that's sort of the poster child of genetics in drug development. Excuse me. It's recently been released. It also lowers cholesterol levels. So it has the property, what you can see here is there's only a red circle. So changing that, the genetics suggests that modulating that gene only changes one kind of thing, heart disease. The next is a gene called CARD9. That's actually also interesting for inflammatory bowel disease. It's something that's being pursued as a possible therapy there. This is the glucokinase receptor. This kind of colored set of wheels means the genetics says that if you mess with this gene, all sorts of stuff happens. So it would say if you're a drug company, probably don't go near that. This next gene, PLA2G7, so this 0 here means the genetics suggest that if you change that, you won't see any difference. That's another interesting example. There's something else I've talked about, LDL cholesterol. Something else you can measure in someone's blood is something called LPPLA2. And it had been observed epidemiologically that individuals with higher levels of LPPLA2 had higher levels of cardiovascular disease. Again, you can't tell from that whether it's causal. If it were causal, it would be worth intervening to lower levels of LPPLA2. Now, we didn't know then whether it was causal. One drug company, GSK, thought it was worth having a go. So they developed a drug which targeted this gene. This is the gene that produces LPPLA2 to reduce levels of LPPLA2. What would we have said if they'd come to us and said, what do you think would happen if we intervene and reduce levels of LPPLA2 via targeting this gene? We would have said, probably nothing's going to happen. Well, we weren't around, and they didn't ask us. They went ahead and did the experiment. They spent $880 million to do a clinical trial to see what happened when you lowered levels of LPPLA2. What did they find happened when you lowered levels of LPPLA2? Nothing. So that's an example where the genetic evidence would have stopped a very expensive clinical trial. So now I want to swap again and give you a sense of the other thing I promised. So those examples are about using genetics to understand how to develop better drugs, which targets to choose, and what the consequences might be of targeting those. Now I want to switch gears and say a little bit about individual variation and how we can make more sense of that with the range of data that's becoming available, not just genetics. So if the technology works. This is an iPad, and I'm about to show you an app that's on the iPad, if it all works. Great. Oh. Yep. Okay. So this is something, again, that we've developed, taking forward our academic ideas in the Spinout company. Not really as a product as yet, but just to give an illustration of what might be possible. So the background here is, Simon mentioned a resource called UK Biobank, which is a study of 500,000 people in the UK on whom there's a lot of information, including information, it's an anonymized study, but information about their medical records and about genetics and many other things. So we took that as an example of what healthcare systems will look like in the future when all of that sort of information is available, and we used it for a number of purposes. So these are the data that's being used in the app is data on real people. We've made up names for them. We don't know their actual names. They're anonymized. So for example, what this screen shows is for one individual, the one that whose name we've made up as Natalie, there's information which is accurate about her age, sex, height, and so on. And then what's shown here are predictions based just on genetics of what diseases she's at particularly increased risk of in the top portion and at decreased risk of compared to the average in the bottom portion. And she happens to be at higher risk of some disease based on her genetics than others. And actually if you can see these grayed out histograms, that's kind of the population distribution of risk. And you can see obviously in these examples, this individual's at high relative risk and in some cases high absolute risk as well. Now, one of the key points I want to get across, most people, when they think about precision medicine and think about genetics and genomics and knowing someone's DNA sequence, they think in terms of prediction. If you know the DNA sequence, you'll be able to say that person is going to develop type 2 diabetes, this person is going to develop arthritis, and so on. We're not very good currently at predicting outcomes based on genetics. We'll get better at that as we get more data. But a key message I want to get across is it would be crazy to do prediction just based on genetics, because there's all sorts of other stuff we measure routinely in medicine, and we should use that for prediction as well. And I'll come back to that in just a moment. But you can scroll through individuals in the app and look at their differing degrees of risk for different conditions and so on. I'll move on to show you something else that we think is potentially helpful in healthcare systems. So this is something which is called chromosome painting. So here are the 22 chromosomes, not the X and the Y chromosome, for a particular individual whom we've called Hector. And it's relatively easy to look at broad scales in someone's genome and across their chromosomes to say whether that piece of, so all of the stuff that any one of us has, we've inherited from our parents and grandparents and so on, and you can trace those back in the past, and it's relatively straightforward to predict for large chunks of people's chromosomes whether those chromosomes most likely come from an ancestor with European ancestry or with Asian ancestry or with African ancestry. And then what you can do, having done that prediction, you can then kind of color in the chromosomes. That's what's shown here. And this individual has a genome which is fairly mixed. So the kind of light bluey color is European ancestry, the greeny color is East Asian, and the brown color is African ancestry. And they've got ancestry from all of those parts of the world. This individual, again mixed, but in this case largely European and East Asian. Someone who's mainly European. These are all UK individuals, so we'd expect a lot of European ancestry, mainly European. This individual is also mainly European. This individual is actually really interesting. We don't know anything about them because of the anonymization, but we do know from their medical record they suffer from something called sickle cell disease. So that's a condition that's endemic in areas where there's malaria, in particular Africa and some parts of Asia. It's not something that's prevalent in the UK. So this is an individual who self-reports as Caucasian, so white European. We don't know, but I can just imagine the clinical odyssey they would have had as a young kid who had a range of symptoms. Probably a typical primary care physician in the UK wouldn't recognize the symptoms of sickle disease because it's not something they have to treat very often. If they did, they would probably rule it out because the person has clearly European ancestry. Eventually it was diagnosed. If you look, you can see just on the end of chromosome 11 and in one other place, there's a tiny bit of the chromosome where their ancestry is African rather than European. Now that happens to be exactly the part of the genome where the beta-globin locus is and where the sickle mutation is. So had the doctor had this available, or probably more realistically, had there been a clinical support tool which was using this information, it should have substantially weighted the possibility of sickle disease and potentially suggested it in light of the symptoms. So, as I said, this is an individual who had no idea, as far as we can tell from their self-report, that they had African ancestry. Probably they had one ancestor with ancestry from that part of the world 5 or 6 generations ago. I want to go back to what I was saying about, about prediction. As I said, when people think about genetics, the first thing that most people think of is prediction with precision that we don't have now and probably will never have about future diseases. But the main point I want to make is it would be crazy to do prediction of future disease risk just based on genetics. What we should be doing is using genetics and all the other stuff we measure in the clinical records. And that's what we did. UK Biobank allows us to do that. There's information collected on individuals of the sort that would be collected in healthcare systems, and information about hospital admissions. So we kind of played a fairly standard statistical game. We've actually got data on these individuals up to 2017. We pretended we only had data up to 2012 and used that to predict what would happen over the next 5 years. You take some of the individuals, there are 500,000 in UK biobanks, so you take some of them and use that to fit a statistical model, use a machine learning type algorithm, and then you take other individuals and you use that to check that you're not overfitting. So we did sensible statistical things. This is actually, in some sense, the simplest of those prediction models that you could do. And again, for each of the individuals, you can say, using all of that information, what are the conditions at which they're of increased risk? And Hector, again, is at increased risk of MI, of a heart attack. And you can even look, in his case, to see what are the risk factors he has that's increasing his risk, and which are the ones that aren't so relevant to him. We've even used the idea of colouring those risk factors, because from a clinical point of view, some risk factors are important for prediction. If you're male and 70, then you're at higher risk of an MI than if you're a 50-year-old female. But those aren't things the doctor can do anything about. There are other risk factors where intervening would be important, and again, that comes back to causality. So you can look not just at individuals, but you can look across a whole cohort. So these are those predictions across all of the individuals. And if you want to, so that might be interesting from a public health point of view. You could say, who are the individuals at increased risk, and have a look at them and pick out their individual risk factors. As I said, we're developing this, I think, so we could get our own sense of how well we can do even with simple prediction methods. As we get much better at keeping medical information and using that and developing the right sorts of techniques, these things, I think, will become much more a part of healthcare, and in particular of personalised medicine. Thanks very much.
It will be Q&A time again.
Peter, it's very exciting to see such a system is being under development. But as you pointed out in your last statement, that to make such a system work, you first of all have to have quite a sizable population. And I think probably you need to have people from different ethnicity or different background in order to have the generalizability of your data. And secondly, it depends very much on how much information that you can gather from the patient's clinical file, and how long you follow up so that you know the outcome in your prediction of the 4,000 or 5,000 phenotypes that you have. So, so I would say this is a project forever in the development and evolution. How and when you think the system is mature enough to be really applied to drug development or prevention of diseases, that I mean, you mentioned a couple of genes that may have either one phenotype or many phenotypes being affected when the gene is perturbed. So when would that actually you become confident that this outcome is actually predicting the correct thing?
No, it's a really good question. Let me take it in two parts. One of them is the bit about drug development and choosing targets, and the other one is about the kind of individual prediction. There's already evidence from two studies, one from GSK and one from AstraZeneca, which shows that even rudimentary genetic information in support of a particular target doubles the success of that target in clinical trials. So that changes the 10% to 20%. And you may think 20% is not very good, but actually just that change would radically alter the economics of drug development. Now, I think the setting we're at the moment, we're already able to be much more sophisticated than that. So I think those sorts of genetic ideas to find or validate drug targets are ripe for use now. And in fact, our company has an announced collaboration with Biogen, a large biotech pharma, to find new targets in multiple sclerosis for them. We're at the early days of that, and I think these approaches, and I think in particular the ones we've developed, can make a big difference. There'll be a point in the future, maybe it's 5 years off, maybe 10 years, when no one would think of developing drugs without this kind of information at their fingertips. because it gives us a way of learning about what happens in human biology in humans. So I think that part's pretty easy on the prediction side. Yeah.
What about people from different ethnicity? We talk about potential collaboration between the 3 cities or the 3 universities. Do you think if we can gather the biobank material from Hong Kong or Macau combined to what you have, primarily English patients, would enrich your database and make it more generalizable?
Absolutely. Those sorts of studies would. China Kadoorie Biobank is another very good example where there's a lot of potential there. I think one point to make is that when, in the first part of my talk, where we're talking about developing drug targets and working out what the typical response is, you might learn something in a Caucasian population that could be helpful for developing drugs that will work everywhere in the world, or you might learn something in an Asian population that would help develop drugs that would work in a European context, because we're learning about the consequences of genetic changes on human biology. I think, exactly as you said, Joseph, when we come to thinking about tailoring this to individual, whether it's clinical support tools of this kind or prediction, that's a much harder problem. It's one where variation across groups, because of ethnicity, because of different, through genetics or different cultural factors, diet and lifestyle and so on, will be much more significant and much more important. So I think that's much further away. I think we'll get to some stage in the future where these sorts of techniques, all the data that's collected, and statistical or machine learning approaches will start to be useful clinically. When that day is, I'm not sure. 5 years, 10 years, 15 years?
But if a chimpanzee's gene is only 1% different from the human gene, how different is the British chimpanzee from a Chinese chimpanzee, or a British man from a Chinese man?
Well, it's interesting. So the genome's the same, and almost all the genes are the same, and you'll know, and maybe many in the audience will know, actually the set of genes we have is very similar to the set of genes that a mouse has, and it's not that dissimilar from the set of genes that a fly has, and it's got quite a lot in common with the genes in a yeast cell. So that's like it's an analogy that's been used before, that's the building blocks. What makes us different from yeast, flies, and chimpanzees is the ways in which the building blocks are used and how they're turned on. So that's all of the machinery about regulation. And that's clearly the stuff which differs significantly between different species and between us and chimpanzee. Again, I think the key idea is this idea that if we use genetics, we can, it's been called nature's clinical trial, we can look at genetic variants which have particular effects on our human biology. And then if you have the right kind of data, you can see that if you change this A to a T, it increases expression of this particular gene. That changes the level of that protein. You can also see changes on other proteins, so they're probably things that are happening downstream in pathways. Some of those we'll already know about, some of those we won't know about. You can then see consequences on other molecular phenotypes, cellular phenotypes, all the way through to biomarkers and disease outcomes. So I think those things will be helpful, and much of that is learning about fundamental human biology. And my claim would be we can potentially learn about that in an Asian population and learn things that will be useful for all humans, or learn about it in a European population. There'll be some genetic variants which are more common in Europe, so that approach will work more easily there. There are others that will be more common in this part of the world, and it will work more successfully here.
Okay.
Peter, I think one of the complexities is that oftentimes with gene mutations, we don't know if it's going to be a loss of function mutation or a gain of function or no change or a de novo function. So for these SNPs that you're using, is that known? Is it known what direction it goes in? And does it even matter?
It absolutely matters. And I mean, it matters anyway in terms of our understanding of biology. And if you're developing a drug, you need to know whether you want to inhibit or agonize or antagonize the target. So direction absolutely matters. Most common genetic variants which have an effect on biology have an effect not through changing the protein which is produced, but they have an effect through affecting regulation of the gene. And as we develop the right kinds of information, so information on gene expression and so on, we're getting better, actually both experimentally, we're getting much better at studying gene regulation directly, but also computationally, we're getting much better at making those predictions. So it's not true in the case of every variant, but there are already enough variants where I can be confident that if you change this A to a T at this position, then in these tissues, it will increase expression of that gene, and that'll have these consequences on proteins and so on.
Okay, I think this will be our last question. Yes.
Hi, Professor Donnelly. Thank you very much for such an inspiring talk. I want to ask if you discover an SNP on a non-coding region of the genome, a non-promoter region, and what would be your recommendation to in terms of regulating what they can do to combat this particular change if it is a non-coding SNP?
So I think I understood the question. Let me try and answer it, and then you can tell me if I've answered the wrong question. So most of these genetic variants, most common genetic variants, have only small effects on people. So the example I showed you on cholesterol levels, that RS12916, it's close to my heart, so I remember its number, RS12916, that has a small effect on the expression of the gene, and it has a small but measurable and real effect on cholesterol levels. But in order to see that effect convincingly, you need to study 50 or 100,000 people. The effect on cholesterol is small of that genetic variant. It's useful, I think, because it can tell us what happens if you perturb human biology in the same direction as the drug. The drug, statins in this case, has a much bigger effect on the gene. That has a much bigger effect on cholesterol levels and in turn on heart disease. So if someone happened to have the C rather than the T, or whatever the example was, variant at that position, you wouldn't be worried about them as a clinician. That will have a very small, so that having the C rather than the A or whatever at that position will have a tiny effect on their individual cholesterol level, and not in a way that would increase heart disease risk. So you probably don't want to try and intervene to correct that in that person. I mean, there are hundreds of these common variants across the genome now known to affect LDL cholesterol. If you had the wrong version of the variant at many of those positions, then collectively they might start to have an effect on cholesterol levels, and you may want to intervene. You'd probably be, depending on their age and so on, you'd probably be monitoring cholesterol anyway, so you would have done it empirically on the basis of the cholesterol level rather than on the basis of the genetics. Did I understand the question? Yeah.
Thank you.
Okay.
Okay, so with that, I think we need to close this session. So let's thank Professor Peter Donnelly in this usual manner. So following this session will be the tea break, I suppose. Yes.
Yes, I believe that we can abandon our ID card, but we never have to use the fingerprint no more, because this is already Stone Age. But please don't forget your iPad, or at least your cell phone should be anytime with the battery. Otherwise, you will be wiped out by the modern technology.